Well, welcome everyone, and welcome Rick. Thanks for coming, even online, to our seminar series. So this is our blood and transfer research unit in blood and health and behavior seminar series. This is a seminar series that we usually run with different speakers online and on site. So this time we have online, and it's really a pleasure to have Rick Kaufman, professor of pathology and laboratory medicine, the Geisel School of Medicine in Dartmouth, US. Well, I don't think Rick need any introduction. Current editor-in-chief of transfusion, a lot of fundamental, essential, key research in this area. So, Rick, up to you. And I think people can ask questions at the end, but it will be as well put the question either raising their hand or actually putting the question in the chat, and I'm sure that Rick will be able to address all of them. Good.
All right. Well, thanks a lot for the invitations. Good to see some familiar faces as well as people that I have not met before. Are you able to see my screen?
Yes, we can. Yes.
Okay. So just to start out, I know that apheresis platelets are used in many of the places where you guys are, including England. So in the US, there's about 2 million platelet collections done per year, and about 95% of them are done by apheresis, which, as you know, just involves connecting a donor to an automated centrifugal instrument. Whole blood goes in, gets separated, and platelet-rich plasma gets pumped up into a bag, and it's possible to collect up to 3 apheresis units of platelets in a single donation, and then red cells and some of the plasma goes back to the donor. And the technology for this became available in the 1980s, and immediately people started to worry that, well, could you make people ill if they donated a lot? So there was concern that maybe you would remove too many platelets and put people at risk for bleeding, or maybe you'd take out too many lymphocytes, and could you make people immunocompromised? And so this is a study that was published in 1981 by Ron Strauss's group. And what they did was to enroll a small set of healthy men who were in their 20s, and these people had never donated platelets before, and they simply had them donate platelets once a week, 10 times basically over 12 weeks. And for each donation, they measured their total lymphocyte count. And what's plotted for each of the platelet collections, so 1, 2, 3, and so on, is the difference for each of the donors, higher or lower, the percent difference in their lymphocyte count at that donation relative to the donor's mean lymphocyte count across the entire study. And so what you can see is that overall, there was a drop in lymphocytes that they saw in this group of about 22%. The investigators felt that this was statistically significant, but not necessarily clinically significant. So anyway, this was an early indication that lymphocyte removal could potentially be an issue with platelet collection by apheresis. But then over the ensuing decades, the technologies got better and people started worrying less about loss of lymphocytes, white cells, during platelet collection. And Ron Strauss was quoted in an editorial in Transfusion in 2008 saying that donor lymphocytes collected or lost during platelet pheresis are so few that the risk of significant lymphopenia and or immune dysfunction is nil. And that remained true until 2017. I came into our hospital blood bank, and there was a phone message that was left by one of our donors that said, "I've been diagnosed with AIDS." And the donor was actually a frequent platelet donor. He had donated 212 times at our hospital-based donor center. And I called him up, and he felt fine. He'd gone bicycling for 20 miles a day before, was in good health, and of course had tested negative for HIV by nucleic acid testing and serology 212 times. So I wasn't worried that he had an infection, but it turned out that he had volunteered as a research subject at an institute called the Ragon Institute in Cambridge, Massachusetts. And there they study AIDS. And so they did a CD4 count on him, and his count was 186, which is below the 200 threshold that's used to define AIDS in people who are HIV positive. And the normal range for CD4 is something like 500 to 1500 cells per microliter. So this was quite low, and I couldn't really tell him why, but he decided to come in and see a hematologist at the Brigham where I was working at the time, and they did some testing on him. And in the meantime, that weekend, I asked our donor center to do CBCs with differential on every donor that came through, both whole blood donors and platelet donors. And it looked like there might be lower lymphocytes overall in the platelet donors. And so I ended up doing a study with John Gansner, this hematologist that our index donor went to see. So we did a cross-sectional study of TREMA platelet donors. So we used the TREMA instrument by Terumo. At the Brigham/Dana-Farber Donor Center, there are many donors who donate as frequently as possible, which in the US is up to 24 times per year, which is true in lots of countries. And so there's a group that calls themselves Team 20, and they donate at least 20 times a year. So we enrolled 20 of the Team 20 people. As a control, we enrolled people who were new donors. So they had donated at most one previous time in the prior year, and then were coming in for another donation. And then we decided while we were doing this, we'd enroll 20 people who were sort of in the middle. So donating 3 to 19 times in the prior 365 days. And for each of their donations, we measured CD4 and CD8 counts, and also collected cells from the little LRS cone that catches white cells in the system. I'll talk about that later. And this was the main result. So there seemed to be a dose-dependent relationship between the frequency of donation and donor CD4 counts. So on the bottom, on the x-axis, you can see that there's the 1 to 2 donations group, the intermediate 3 to 19 donations group, and then the frequent donors 20 to 24 times in the past year. And the more frequently you donated, the lower your CD4 count tended to be. The little dotted line at the bottom is a CD4 count of 200, the threshold for AIDS in people who are HIV positive. And there were 2 out of 20 donors, or 10% in the intermediate group, that fell below that threshold. And it was 6 out of 20, or 30% of our Team 20 donors. And the blue symbols just indicate that at some point in the donor's career, they donated 20 times during 1 year. Some of these donors have been donating for many years. Now, there was one major limitation to this study, and that is that it turned out that the new donors tended to be younger. So this is the median ages are shown here, 38 versus 61 or 63 in the intermediate or frequent donors, respectively. This kind of makes sense in that many people who donate 24 times a year are retired. It takes a lot of time to be able to donate. And this is important because, of course, your peripheral blood T cell counts decline with age. So all T cells, CD3 positive cells, are shown on the left, and you can see kind of a downward trend with increasing age. And the same holds true for CD4 T cells shown on the right. So it's not like your T cells fall off a cliff as you age, but people in their 60s tend to have lower CD4 counts than people in their 30s. And so that was automatically going to confound the results of that initial study. So I'm just going to switch gears for a second. There are 2 main instruments that are used around the world these days to collect apheresis platelets. So one is the TREMA XL, and one is the Fenwal Amicus. And they both work by the same principle, but there are some important just engineering differences. They both produce leukoreduced platelet units. But the TREMA instrument uses something called the LRS, or leukoreduction system chamber, and the phenylamicus has no LRS. And the LRS, or leukoreduction system chamber, is just a little plastic cone that is designed to catch white cells and keep them from ending up in the platelet product. It looks like this, and platelet-rich plasma kind of percolates through the pointy end and out the top. And what happens is a dam of white cells gets formed near the top, and platelets kind of percolate up through that and end up in the collection bag. And it turns out that this system is quite efficient at catching white cells. When we looked at what was in these little plastic cones for the different groups of donors, what's shown in blue are the 1 to 2 donations group, red is the 3 to 19, and green is the 20 to 24. You can see that lots and lots of lymphocytes are caught by this thing, as well as monocytes. So we think it's about 15 or 20% of the circulating lymphocytes and monocytes that are trapped during a single platelet donation. For whatever reason, neutrophils are not caught by this system very well at all, nor eosinophils. Some basophils end up trapped by this cone. And one side benefit for using this exact instrument is that these cones then contain viable lymphocytes that many immunology basic science researchers like to use. So lots of places will give or sell these cones to basic scientists to use for experiments. So it did appear, though, that donor lymphopenia could occur in the absence of an LRS. So this is a study that Norbert Ehren's group did in Germany that was really good. So they looked at donations during a 4-year period where their donor center was using the TREMA. And they found a relationship between donation frequency, so sort of going from left to right on the x-axis, as well as total, that is cumulative platelet donations. So the more green symbols mean you've donated less over your lifetime, and the more red, more donations, up to 200 or so. And you can see that both with more cumulative platelet donations and with more annual platelet donations, sort of heading toward that bottom right-hand corner of the plot, the total lymphocytes tended to be lower. So that was consistent with what we found. But they also looked at a time period of the same length, so another 4-year time period that was earlier, when their donor center had used the phenylamicus instrument. And there, you can see the data are a little messier. The relationship's not as clean, but it still looks like there might be a relationship with lower lymphocytes with increasing platelet donations or increasing cumulative donations. It's just not as strong or as clean as it is with the TREMA. And so to try to resolve all this, I ended up doing a study with the BEST group. We were calling this the BEST PAL study for platelet-pheresis-associated lymphopenia. And so we wanted to address kind of the major limitations of the previous studies. And the main thing that we did was to enroll 126 age and sex matched whole blood donor controls. So that's symbolized by the black symbol on top. And we collected at a single visit a blood sample for CD4 and CD8 testing. We also enrolled 77 frequent amicus platelet donors, shown in green. And at the bottom, shown in blue, we enrolled 131 frequent TREMA donors. And of those, 91 were collected without the use of plasma rinse back, which I'll talk about later, but 40 of them who were collected at sites in English-speaking Canada were collected using the TREMA instrument, but also doing this plasma rinse back optional procedure that nobody else was doing. And this study was done at 31 donor centers that were in the US and Canada and Australia. And shown on the right, for a subset of the frequent amicus and TREMA donors, we did have some samples collected longitudinally at 3, 6, and 12 months to kind of see what the T-cell subsets were doing over time. And so this was the characteristics of the participants at baseline, just at their enrollment. So just the top line is showing the numbers. So 126 controls, 77 amicus donors, and 131 trema donors. And you can see that the ages were pretty similar, that the amicus donors happened to be a bit older with a median age of 66, and the whole blood controls and the trema donors were 58 and 59 respectively. Just about everyone in this study was male. And for the whole blood donor controls, we wanted people who had really not donated very much at all so that they would be sort of pristine. And so they could donate up to 6 times in their life. The median number of lifetime donations was only 2, and they could not have donated any whole blood in the prior 6 months. The amicus donors, in contrast, had donated at least 20 times, but the median was 116 times lifetime. And same with the trema donors. So to get into the study, you had to be a frequent donor, donate 20 times in the past year. And they had donated a median of 137 times. And we had one donor from Stanford who had donated 759 times, which takes decades to do. And so if you look just at white cells among these 3 groups, so whole blood donors, amicus donors, and platelet donors shown on the right, you don't see much of a difference. The trema platelet donors actually had slightly lower white cells, but it's not anything you'd ever pick up routinely. But as we expected, we started to see some interesting things with the CD4 counts. And so you can see that the amicus platelet donors had significantly lower peripheral blood CD4 counts than the whole blood donors, and the trema donors had significantly lower CD4 T cell counts than the amicus donors. The dotted red line or dashed red line is showing where a CD4 count of 200 is, and there were about 4% of the amicus donors that fell below this 200 threshold, and it was about 10% in this study for the trema platelet donors. And we only saw people who were below 200 in this study if they had donated at least 200 times lifetime. And the pattern for CD8 T cell counts was really similar. The dashed line here is just showing the lower limit of normal for the lab that performed the testing, which was 125 cells per microliter. So particularly in the trema donors, there was a very large number of them that had abnormally low CD8 counts. So I mentioned this rinse back business. It turns out that with the trema instrument, it's possible to flush some of the cells out of the tubing and return them to the donor. Now, it doesn't touch anything that's in the leukoreduction system cone, so nothing comes out of that. But you could imagine having some red cells or white cells trapped in the sterile plastic tubing, and so by flushing back some plasma at the end, it takes a few minutes. It is possible to leave the donor with, in theory, a little bit more cells. In 2013, Dana Devine and English-speaking Canada decided as a policy they were going to do this rinse back for all their donors. They weren't thinking about lymphocytes at all. They wanted to help preserve hemoglobin, to help preserve the red cells in the donors, and so they were flushing everybody back. And so we compared the 91 trema platelet donors in this study who did not receive any rinse back with those from the Canadian Blood Services sites, of which there were 40 that were getting this rinse back. So the median age was exactly the same, 59. Again, this is almost exclusively men. And lifetime donations were a little bit different, but actually higher, 157 versus 121 in the donors receiving rinse back versus not. And this figure just shows that how the proportion of lifetime donations that a donor may have made, receiving rinse back or not receiving rinse back, kind of depended on how long the donor had been donating. So each row in this figure represents a single donor's career. Donor number one had donated about 400 times. The first more than 200, shown in orange, were done in the period before 2013, before rinse back was implemented. in Canada, and then another about 200 donations shown in blue were done in the period after. And then there were donors at the bottom of the figure who started donating after April of 2013. So all their donations were done with them receiving rinse back. So it was kind of a mix, but you can see that just visually that most of the donations in this rinse back group were done using rinse back. And it seems that plasma rinse back at least appears to partially mitigate against lymphopenia. So the whole blood donors are shown on the left. The treatment platelet donors not receiving rinse back are shown in the middle, and the treatment platelet donors shown who receive rinse back are shown on the right. And what's plotted here is the CD4 count at enrollment. And you can see that there were plenty of donors in the no rinse back group with CD4 counts below 200, and there were just none in the donors getting rinse back. And the median, as shown by the box plot, was also higher among those getting rinse back. And the same pattern was seen with CD8 cells as you'd expect. So we did both univariate and multivariate analysis for CD4 T cell count. And the way to read this table is for each of the risk factors that's listed, what the number shown basically tells you how many CD4 cells on average to add or subtract to your peripheral count. So for the univariate model, for instance, if you happen to be collected during your donation career on the Amicus instrument, you can add 172.7 peripheral blood CD4 T cells versus the TREMA. So the TREMA seems to, as we've seen, take out more of the CD4 cells, or is associated with a lower CD4 count, I should say. For each lifetime donation you lose about 1.3 CD4 T cells. And for each year of age, though, you lose about 6. And then you can see that those held up in the multivariable model. And the rinse back also seemed in the univariate analysis to be associated with having 139.4 more CD4 positive T cells per microliter. This was not included in the multivariable model because not everybody got the rinse back exposure. So is this harmful? Is PAL or platelet-apheresis associated lymphopenia harmful? So still a little bit unclear, but so far it looks okay. This is just a study that was done using the NHANES database. This is about 32,000 people representing kind of a typical US population. There were about 20% that had an absolute lymphocyte count below 1,500 and about 3% that had an absolute lymphocyte count of about 1,000. And you can see that the hazard ratio for all-cause mortality goes up with lymphopenia. So this is looking at sort of broadly at the effect on a unselected community of having lower lymphocytes. So our colleague Gustav Edgren in Sweden, using the SCANDAT3 database, tried to see if he could see an association of platelet-apheresis-associated lymphopenia with infections. And so they published a really challenging to read paper that I'll just talk you through really quickly. So using their database, they were able to look at something like 26 million person years of exposure, both of patients and platelet donors. And in their database, they found about 75,000 platelet donors, of which only 95 actually had donated more than, had donated 20 or more times in the past year. So there's not a lot of very, very frequent platelet donors in Sweden and Denmark. I think we tend to push people quite a bit harder in the US system. Anyway, looking at an exposure window of 10 years, excluding the most recent year to try to prevent confounding by the healthy donor effect, they did find that immunosuppression-related infections did seem to go up with more donations. And the same seemed to be true with common bacterial infections. Now, I will say that, and what they were comparing was donors who donated platelets or plasma using an LRS or not using an LRS within their database. Now, what I will say is they did not see a lot of awful infections. So most of the immunosuppression-related infections, and their event rate was really low, were things like shingles, zoster, and so nothing like disseminated mycobacteria or something you might see in full-blown AIDS. So anyway, while intriguing, this is still a retrospective study subject to some confounding, and nothing terrible was seen in terms of potential for donor harm, but still a little concerning. In the original study, we looked at T cell receptor diversity by doing immunoseq assays on lymphocytes that were left in the TREMA leukoreduction system chamber, and we didn't see a difference in T cell receptor diversity among our infrequent, intermediate, and frequent donors. So that was reassuring. And in the Best Pal study, we looked at IgG levels. And so whole blood donors are shown on the left as before, and the amicus donors are shown in the middle. The TREMA donors are shown on the right. And you can see that the IgG levels were a little bit lower among the platelet donors versus the whole blood donors, actually lowest in the amicus group. But this is a little bit to be expected in that there's a fair amount of plasma that the donors would be losing with each of their donations. They're donating at least 20 times a year. And so like with frequent plasma donors, you expect to see some decrement in IgG. And I think that's what we observed. T-cell subpopulations were looked at in the frequent donors in our original study, and I won't spend a whole lot of time on this except to say that we used a whole lot of flow markers, made a couple observations that I can't really interpret very well. So it looked like naive CD4 T cells were somewhat lower in the very frequent donors, the 20 to 24 group, but that effector memory CD4 cells were somewhat higher. And I don't know what this means, except to say that it's reminiscent of something called LIP, or lymphopenia-induced proliferation, which is something which can be seen in animal models. So if you remove lymphocytes from a mouse, sometimes what can be observed is that their T cells, without ever being exposed to any sort of antigen, will undergo some surface changes to look like they're becoming more like effector memory cells, but in the absence instead of any real exposure to a pathogen or any sort of antigen. So anyway, are these donors having LIP? We don't know. A couple of reassuring studies were done using vaccination. So this was done by Renee Bozen's group in Quebec during the pandemic. And what was done was to take frequent platelet donors, there were 27 who had a platelet count that was, CD4 count less than 400. That's shown by the green symbol on top. And there were 16 that had CD4 counts that were at least 400, shown on the bottom. And at a couple different visits, these donors received mRNA vaccines. And the bottom line is that independent of what your CD4 count was, it was possible for these frequent platelet donors to be able to respond normally and to make anti-receptor binding domain IgG. So as I said, that was a reassuring one. At Dana-Farber, we did a study that was similar. So we took several donors who donated a range of different times. So some really frequent donors and some new donors. And we enrolled them in a study where they were given this meningivax, this vaccine that's often given to college kids now, but wasn't given years ago. And so for most of the donors, this would have been a brand new antigen. And so it's a quadrivalent vaccine against N meningitis. And so what's shown are the seroresponses to the 4 different antigens that are included in this vaccine. And it is supposed to be T helper, that is CD4 T cell dependent. And you can see that most of the donors had a good response to vaccination. So if there's a a yes on the bottom, basically the plots on the right in each of the 4 sets, means that the donor had a reasonable antibody response to the vaccination. And you can see that the CD4 counts are kind of all over the place. There were some with really, really low CD4 counts from having donated platelets a lot, but they still responded just fine to serogroup A, C, W, and Y, the 4 antigens. So again, this is at least showing that their immune systems, insofar as it's needed to be able to make antibodies to a new vaccine, seem to be good, even if you had a CD4 count that was kind of scarily low. One thing we did find is that this lymphopenia associated with platelet donation can last a really long time, even after you stop donating. So I had a fellow who sent 50 letters to former frequent platelet donors. So these were people who used to donate at least 20 times a year, but then stopped donating for whatever reason. They moved, or I don't know, maybe some of them got sick, or some of them just didn't feel like donating anymore. And there were 15 responses, and these people were willing to come in and provide a sample. And so again, we looked at CDVCs and CD4 and CD8 counts. And what's plotted are CD4 counts of current platelet donors versus our 15 former platelet donors on the right. You can see there were 2 of them who still had CD4 counts below 200, even though they'd stopped donating a while ago. So one person had stopped donating for about 2 years, and one had stopped donating for about 1 year, and still their CD4 counts remain below 200. So I'm just going to wrap up here. In conclusion, we found that independent risk factors for platelet pheresis-associated lymphopenia include age, lifetime donations, and the apheresis instrument. PAL occurs both with the TREMA, which has an LRS, and the Amicus instrument, which doesn't, but it's more frequent and seems to be more severe probably with the TREMA. In the BEST PAL study, CD4 positive T cell counts less than 200 per microliter were seen in donors with at least 200 lifetime donations. They were seen in about 10% of frequent TREMA donors and 4% of frequent Amicus donors. And they were not seen among frequent TREMA donors that receive plasma rinse back. The mechanism for PAL is unclear, but on balance, we think that bulk lymphocyte removal probably contributes. To date, there's actually no definitive evidence that PAL is harmful, but it can certainly persist. So recommendations. We think when platelet donors are consented, they should be notified of the risk of lymphopenia. There were a couple of cases where, even though they felt healthy, there were platelet donors we found who were incidentally noted to have lymphopenia. And so in at least a couple of cases, donors have undergone bone marrow biopsies that didn't find anything abnormal, except that, they probably had PAL, and this led to possibly testing that they likely did not need. And finally, we think that when collecting platelets with the TREMA instrument, that plasma rinse back should be used to help try to preserve the lymphocyte count. So just to finish, I think that frequent platelet pheresis perturbs T cell homeostasis. We don't know exactly what's going on, so I'm just going to show you a really crude cartoon that I made. So this is, as I said, quite crude. So this is a simplified version of normal T cell homeostasis in adults. So each of the little blue dots represents 10 to the 9th T lymphocytes in your body. And in the middle, the circulatory system is shown, and around the periphery in gray are the immune system organs. So I'm showing the thymus, the spleen, the bone marrow, and lymph nodes. And the number of T lymphocytes is roughly proportional to what's actually in a person. And so we think that T cells are first in equilibrium between some of these immune system organs and the blood, as shown by the teal arrows going back and forth. But also, it's really important to note that most of the T cells that are in your body are not in your blood. So it's only, in fact, about 2 or 3% that are circulating. And the vast majority of your T cells are in the lymph nodes, bone marrow, spleen. And And we actually don't know like what exactly T cells are doing in your blood. They may just be commuting to work. We think that sort of all the activity, antigen recognition, expansion, and so on is likely happening in the periphery. And maybe they're just moving around in the circulation, even though we know, for example, that the peripheral blood CD4 count is critical for monitoring people with HIV. It may have little to do with what's happening immunologically. And as shown at the bottom with the syringe, seroresponsive vaccination is normal in this setting. Okay, so we take someone like that and we subject them to long-term frequent platelet pheresis. So now they're donating platelets 20 times a year for 10 years. And they end up with something like this, where we actually have no idea what's going on in their immune system organs. We do see a sustained, reduction by about 1 log of the T cells that are circulating in the blood. And they may be just completely fine immunologically, and maybe they've just hit a new set point. Maybe if you did a lymph node biopsy, you'd find lymph nodes look completely normal. It's also possible that the lymphocyte numbers are lower in the periphery. We just do not know. But as we've seen, the seroresponse to vaccination at least looks normal, which is a pretty good test of kind of at least part of the complete immune system. So so let me stop there. I really want to thank my many collaborators, both with the BEST group and and many other places, particularly John Gansner from the Brigham, Renee Bosin from Hema Quebec, and Denise Marks from Australian Red Cross. And thanks, and I'm happy to answer any questions.
Excellent. Thanks, Rick. Really great overview. If you can stop sharing your screen so we can see each other, I guess you can see as well.
Sure.
Yeah, here we are. Excellent. Thanks. So there was Dave.
Oh, hi, Rick. Can you hear me okay?
Oh, yeah. Hey, Dave.
Yeah, lovely talk. Very clear. When you say there's no evidence of harm, could you just go, perhaps I missed the significance of Gustav Edgren's findings. Do you think most of those were age-related effects? Because at first sight, unless I misunderstood the data, do look relatively harmful. And I mean, shingles is pretty nasty, and I wouldn't say it's without risk either.
So I think what I would say is there's no definitive evidence of harm. I think that's probably the safest and truest way that I can say it. I think that there's some evidence that's suggestive of harm. That would be the Edgren data. And the problem, as I said, is I think it's just too limited. So I think that there were 11 events.
Okay.
And the other thing is they didn't see any in their group that you'd expect to be at the very highest risk. That is the people who collected using the LRS, who had the most collections. And so there's what looks to be a signal there, but I don't think you can be confident that it's right. Now, Gustav will tell you that he's really much more worried than I am about this. But I think it's sort of a limitation of that study. It would be really nice if we could do a similar study to what he did, but using a data set that included a lot more frequent donors like you'd find in the US. As I mentioned, I think we had more frequent donors at my own donor center than they had in their entire study. Only what's great about Scandinavia is everybody gets tracked their whole life. In the US, it's a lot more freewheeling and disorganized. And so there's not a way to do that kind of study. Yeah. Now it's certainly true that we haven't seen anyone who's been a frequent donor who's been sick. On the other hand, there's of course a selection bias in that you have to be well and healthy to be able to donate. So maybe we're only seeing the healthy people. And when we wrote to the former donors, none of them seemed to have anything that you'd be particularly worried about. But that's where it is. Yeah.
Okay, thanks.
Katja.
Thanks. Yeah.
Hi, yes, thank you very much for your nice talk. It's really good to see the progress. I've seen it a couple of times during the BEST meeting, of course, as well. Very interesting. A bit as a follow-up on Dave's question before, there's two things actually that I'd like to ask you. So one is to what extent you think this healthy donor effect also has played a role, because I think these studies, all of them are observational. So you look at one point, so the ones that have donated but became ill, they are simply not in your group anymore. And then a second question is related to the fact that all of the studies like only included or almost only included men and no women at all. So I wonder if you could also hypothesize a bit on what you would expect to find in women. And I also wonder actually why they're not included in those studies, or mainly not.
Yeah, so the first question is, I'm not sure why it was so predominantly men. I will say that in our donor center, there were definitely more male donors than female that would donate platelets frequently, but it wasn't 100 to 0, or even it was more like, I don't know, maybe like 65%, 35%, something like that. So why in the study it came out like that, I'm not sure. So that I honestly don't know. There were no restrictions.
It's not because you've selected like the most high frequency donors or something.
Yeah,
perhaps.
I'm really not sure.
Okay.
I do, as I said, I think the only thing I've seen is that there's definitely more male donors, but it was skewed more than you'd expect, and I'm not sure the explanation for that. As far as the healthy donor effect, yeah, so that's an incredible limitation of all of these studies, except for the one where we wrote to the old former donors, and we only got 15, and so the numbers are terribly, terribly small.
Yeah, that's
So
true,
that
yeah.
one thing that could be done would be a larger scale version of that sort of study. That would be practical to do, I think. To do a prospective study would be super hard, because you don't know who's going to, first it would take, you'd have to have a really young fellow who's going to, carry on after you've retired with such a thing. And you don't know who's going to go on to donate really frequently for a long time. And as far as the healthy donor effect, in the Edgren study, they did about as well as anybody can do, I think, in trying to mitigate against that. Just briefly, they looked at donations as an exposure over a 10-year period, but they left out the last year, so that if you were knocked out by being sick, then that would be okay. You'd still have that exposure in the 10 years before. And they also compared, as I said, people donating the exact same number of times with or without a leukoreduction system chamber. At the time, it wasn't known that you could still get lymphopenic even without a lymphocyte reduction chamber. But yeah, I think it's an issue. I'm just not sure how to address it exactly. Yeah, I feel better in my heart after seeing the vaccine studies, but it doesn't exclude the possibility that there is some harm from this. But we didn't see anything like. So I was worried when COVID started. Are these people going to be more at risk for severe disease? And maybe that would be something that could be looked at, although, again, with our frequent donors coming in, we didn't see it. So,
Yeah, thank you.
Thanks. And any additional question for Rick?
So, sorry, I was going to put a comment in the chat before going. I mean, we could, of course, vaccinate against shingles if there was concern. I must say, I tend to slightly side with Gustav that it's sort of worrying data, but I'm not sure where taking the presumptive position that, we should try and mitigate the risk if we can.
Yeah, well, as I mentioned, I certainly think it's worth telling the donors about. The FDA used to require that, and then I think in 2007 or so, removed the requirement that in the donor consent, you had to tell them that their lymphocytes might become low. And I think that needs to be put back in to at least make the donors aware. And just so, as I said, there's been some scares, if nothing else. And I don't. And anyway, like I said, it's a hard issue to get at just because of how long this takes to develop. So, yeah.
It is.
Rebecca.
Hi, Rick. I had two questions, one of which I sort of asked you by email, so I think I know the answer. But anyway, the first one is, I suppose, well, it's really a comment. I suppose what is, in the absence of sort of definitive evidence, there's two things we could think about. One is the consent issue, but the other is the rinse back process, and whether you just take a precautionary approach in the absence of data, if you know what I mean. So I suppose that's something practical, isn't it, we could think about. And then the other thing is so these are platelet donors, and then you've obviously got the plasma donors as well, which tend to be different instruments and so on. And we're about to hopefully undertake a quite a large study of plasma pheresis donation volume and frequency, which Emanuele is leading. And so one of the things we were thinking about is, what risks do we want to look at and what testing are we going to do and all of that. So and I was thinking about this lymphopenia side of things as you've obviously presented it before in BEST and so on. And in the end, what we've sort of decided to do, but I'd be interested whether you think this is sort of adequate or not, is just to look at a total lymphocyte count to see whether there's any sort of difference in the arms of the study and compared to our whole blood donors in lymphocyte count, because obviously going to the extent of doing CD4s and CD8s is quite difficult, as you know, because you've done the studies. But do you think that's like a good starting place, or would you do something else, or is that reasonable? I think is my question. I think
that's okay. I mean, you can absolutely see a difference in, say, mean total lymphocyte count if you look at a group of platelet donors versus whole blood donors. Everybody gets a CBC, but they just look at the hemoglobin and the platelet count, and lymphocytes get ignored. And so you'll see it if there's it's not hard to see a difference, at least in those groups. With plasma donors, maybe. With respect to the rinse back, I think it's a good idea. I think I was grateful to Canadian Blood Services. They enrolled some extra donors specifically to look at this issue, and it really seemed to make a difference. There were plenty of donors with sort of an equal number of lifetime donations who did not have rinse back that had really low CD4 counts, and it really did seem to matter. Now, Renee Bosin will also give you a slightly different take. She's not sure about this. The company says that the rinse back will return 20 to 70% of what's in the tubing. It's very clear it does not. The company and independent investigators have found it doesn't do anything to what's in the cone. So how the rinse back even works, I'm not sure, but maybe they're cells sticking to plastic. But yeah, I think it's probably that would be a precautionary principle sort of thing that I think would be reasonable. It takes a few minutes. That is, there's not a huge cost to it, both in time or money or anything. And it does seem to matter, although it would be good to have sort of independent confirmation of that. I think Vitalant has at least been talking about doing it in the US. I'm not sure about other donor centers. I don't know.
I suppose it's something that if we considered it, we could maybe kind of assess, before and after or whatever, something to just try and add to the data. But I don't think we do that. I need to check in NHSBT. I don't think we do the rinse back part, but I need to check my facts.
It's very rare. to be done in the U.S. till now. I think because of these data, I think some people are at least thinking about doing it. As I said, it's not a huge cost. Yeah.
Yeah, we have about 10,000 donors, I think, that platelet donors. So yeah, anyway, Emanuele, something to think about.
Yeah.
And then if you do it, do you do it for everybody? Do you do it for the frequent ones? Like
Yeah. It's for us. It will be. I think it will be quite a thought. So I think it's something we need to think about.
Good, excellent. Any final additional question for Rick? If not, thanks again. Fantastic talk.
There's
Very
one
interesting
online.
data. Oh, it's one online. Sorry, I missed that. Yeah.
No,
Cyril,
that's
yes.
just me
Yeah,
being
sorry.
curious about, because your study seems to provide the opportunity for a self-controlled before and after INSPAC. I don't know whether you've thought about analyzing the data that way.
Self-controlled comparison before and after. Yeah, I think you could. So that was sort of what, and I don't know if, I think that if a donor center decided to adopt it at a particular point in time, yeah, I think that kind of study would be worth doing for sure. Yeah.
Good. So, well, if there's no additional question, thanks, Rick. Great to have you here.
Oh, my pleasure.
Virtually, hopefully at some point we'll be here in person in Cambridge.
Next time in person, for sure.
Yeah. Excellent. Thanks a lot, and thanks for everybody attending. I think there's some advertisement as well of the next, not blood donors meeting, but there's another meeting happening on next Monday. So if people want to attend online or on site, I think we'll be here in Cambridge again.
All right.
Thanks.
Thanks very much.